Good evening, children. This is my fifth video on the chapter Mineral Nutrition in Plants. In the previous video, I explained symbiotic nitrogen fixation, the steps in nitrogen fixation in leguminous plants, formation of bacteroids, mechanism of nitrogen fixation, the function of enzyme nitrogenase, and the pigment leg hemoglobin, how it functions in the process. Now, one thing what is remarkable is that overall in this process, we studied how the atmospheric nitrogen is converted into ammonia by certain prokaryotes only, right? These prokaryotes are capable of this conversion because they have an enzyme nitrogenase present in them. This nitrogenase enzyme is sensitive to oxygen, right? So, who prevents it from coming in contact with oxygen? It is a pigment called leg hemoglobin. So, this is what we saw and studied in the previous video. Now, once this ammonia is formed, what will be the further reaction of this? Let us proceed with this today, right? So, this ammonia, when it is formed, we are starting from here, fate of ammonia. This ammonia is protonated into ammonium ions. Ammonia is highly toxic, and for the same reason, it cannot be accumulated in plants. Thus, ammonia reacts with organic acids and it forms amino acids which are carried by phloem to various parts of the plant for protein synthesis. So, the destination of nitrogen fixation is formation of proteins, which is ultimately formed and thus utilized for growth, right? Now, in today's topic, what I am going to start, children, I must tell you that what I have written from here for you all, that is something about Azolla, Anabina, then Azospirillum and Nostoc. This is not given in your textbook. I will show this. This is in your textbook, page number 12.15. We are talking about the role of Anabina, Azolla, and Nostoc under cyanobacteria. Then in leguminous plants, Rhizobium, which I have explained, it's given in your book, right? And this Azospirillum. So these four things, children, number 1, 2, 3, and 4, Azospirillum, right? I am giving you an extra, so you must have your content with you. And in the hierarchy of the flowchart that I had prepared, children, you remember last time I told you the 5 ticks are only in your syllabus. So according to the 5 ticks, Rhizobium I have finished just now. I'm starting with the other 4, right? So ensure that you're writing this very, very carefully. We are starting with Azolla and Anabina, right? Azolla, it's a small water fern. The complete name is Azolla pinnata. The species pinnata, it grows in ponds, ditches, and stagnant water bodies. Now, in this diagram, this is an Azolla leaf. The green colored structure is Azolla leaf, which is found in ponds, ditches. Now, in the cavity of the leaf, you can see there's a cavity. There lives a cyanobacteria called Anabina azollae, right? The complete name is Anabina azollae. When Azolla was only a fern, right? Then Anabina is a cyanobacterium. We also call it as a blue-green alga, right? So this Anabina azollae, this a cyanobacterium, right? This is a blue-green algae, BGA we write in short, or a cyanobacterium, right? This grows as a symbiont in cavities formed in the chlorophyllous leaves of the Azolla plant. All right. Now, why do you think this association is important? This association is very, very important to increase the yield of certain plants like rice. You can imagine as high as some 50% of the yield that is increased. The output is increased by 50%. So, Azolla Anabina symbiosis forms a very efficient biological nitrogen fixation. It is mostly seen as a green manure in rice fields. It is inoculated before rice transplantation and is supplemented with 8 to 20 kg of phosphate per hectare. Azolla may be grown along with rice crop and the use of Azolla Anabina biofertilizer increases the yield by over 50%. So the dash here in between tells you the relationship between a fern, Azolla, and a cyanobacterium or the blue-green alga, that is Anabina. So this is very, very useful for the rice plants, right? So next we come to Azospirillum. Azospirillum is a gram-negative bacterial genus. This is found in non-leguminous plants, right? And it can promote plant growth by mechanisms of tolerance of abiotic stresses. Abiotic means the non-living factors, right? And what these factors can be? These can be named as induced synthesis tolerance, induced systemic tolerance, osmotic adjustment, production of phytohormones, phyto means plant hormones, and defense strategies like expression of pathogenesis related genes. The bacterium grows, that is, lives in soil around the roots of plants like maize, wheat, and rice. It looks like this, rod shaped with a flagellum at one end, right? Recently, the scientists at IARI, this institute is called as Indian Agricultural Research Institute. It's located in Delhi, right? The group of scientists, they have isolated Azospirillum bacterium from the roots of grasses, rice, sorghum, and maize. Simple seed inoculation with this bacterium increases the dry weight of cereals, and when mixed with a chemical fertilizer, it gives high yields. So again, this is very, very important for the growth of cereal plants and acts in increasing the yield. Coming next to nostoc. Nostoc is a cyanobacterium, just like here, Anabina azole. This was also cyanobacterium. Cyanobacterium is also called as blue-green alga. Short form is BGA, right? So how it looks like? It looks like something this. It's got a beaded appearance. A microorganism which is composed of beaded filaments which aggregate to form a gelatinous mass. This grows in water, damp places, and it's able to fix nitrogen from the atmosphere. Nostoc can be found in soil, on moist rocks, at the bottom of lakes and springs, both fresh water and salt water, and rarely in marine plants. So this is nostoc filament. The red colored structure shows there's a heterostis formed at some stage of the life cycle, right? So again, I repeat, children, what I am doing today is the hierarchy table here. The blue ticks I am doing: Anabina, Azolla, Nostoc, and Azospirillum. I have given you extra notes, whereas this is what I have totally explained from the textbook. So why I have done this? Starting from here, I have explained you why I am giving you number 1. Then this is the second, third, and fourth. The reason is here that in your textbook, except these names, there are no details and explanations given, right? Now I must show you what in the syllabus I am teaching. This is page 5 of your syllabus, and I am teaching today biological nitrogen fixation. This we have done. Then non-symbiotic nitrogen fixation and symbiotic nitrogen fixation. Today we have done role of Azospirillum. Rhizobium we have completed last time. Today we are doing rows of Azospirillum, Azolla, Anabina, and Nostoc. So in short, children, the role of these you must be very clear with. Then importance of nitrogenase enzyme, that is a complex which is formed, and leg hemoglobin pigment. Nitrogen cycle, though I just explained you briefly in the previous time, but here it requires only the graphic outline, right? And the same I am giving you as a homework from page number 12.13. So this is your homework for today, children. You will draw this outline of the nitrogen cycle. Details may not be studied into very in-depth knowledge, but the outline can be asked, children. And here you can just generalize how the cycle, it's a regular supply of nitrogen to the plants which is maintained through the cycle. And in this, there's a regular circulation of nitrogen between three nitrogen pools. Number one, the atmospheric nitrogen, then the soil nitrogen, and then the nitrogen present in the biomass, right? Which could be the decaying biomass, plant or animal biomass. So please draw this nitrogen cycle in your copies and practice accordingly. So that's all for just now, children. I'll wind up the class here, and I'm sending you the notes from here, the fate of ammonia, and these extra things starting from here. All these four, please draw and write down carefully for their role in nitrogen fixation. And that's all for just now. Thank you.